It's me again, Madam Jennifer from Educare TZ. Welcome my lovely Form 2 students in our biology session. And in our today's session, we are going to solve the Necta question for biology subject for the year 2017. Welcome. Today's question is question number 5 for the year 2017. And the questions from an A stated that outline 7 steps of scientific process used by scientists in the scientific investigation. Outline 7 steps of scientific process used by scientists in the scientific investigation. The question Roman B stated that why hand lens is useful in biology. Why hand lens is useful in biology. My lovely Form 2 students, before we answer our question, first of all, we have to familiarize ourselves with the direction of a particular question before you attempt any question. The question Roman A needs you just to mention, to outline the 7 steps of scientific process used by scientists in the scientific investigation, just to mention them. Also, the second one is you to explain why the hand lens is useful in biology. There is no limitation of the point here, but you are needed to be brief as possible. Also, before you answer the question, you have to make sure that you look on the marks that has been attributed to a particular question, so that you can have a room to tackle or to attend to the question. So, and the second one, after knowing the direction, so now let us familiarize with where the question has been derived. The question Romans A has been derived from our lovely topic of the scientific procedure. It is a Form 1 topic. In that topic of the scientific procedure, measurement, observation, and scientific method is where this question has been derived. The question Roman B has been derived from our lovely topic of Form 1 2 in that topic of the biological laboratory. So you're mostly welcome to subscribe it at Educare Media so as to widen up your understanding, both at the Form 1 topic. So in order to attempt them, you need to have deep understanding in these two concepts. So let us start by solving the question Roman 1A, which requires you to outline the seven steps of scientific process used by scientists in the scientific investigation. Also, this is one of the familiar things for you. You learn it from primary, the njia za utafiti wa kisayansi. It is called njia za utafiti za kisayansi. In your Form 1, now you are Form 2. Na vile hata Form 4 ili somo linaweza likatoka. So all topic unazisoma kuanzia Form 1, Form 2, Form 3 to Form 4. They are very important for your summative assessment katika your Necta, your final Necta examination. Make sure kwamba kila topic unaielewa, don't ignore, usije kadharau topic yoyote ile. Both are important katika maisha yako na vile vile katika your performance. So, by starting with the question Roman A, for those wale ambao hajasoma topic ya scientific procedure for faida yao, we have 7 main scientific procedures. Kuna identification of the problem, kuna formulation of hypothesis, kuna experimentation, kuna data analysis and interpretation, kuna data collection. Baada ya data collection, kuna data analysis and interpretation, kuna drawing the conclusion as the last one. We have 7 major points. Kwa mtu kuweza kwa you just need to mention them like that. Lakini kwa ambao jeza kwa 20, I would like to provide you a brief explanation so to see what is going on how you can attempt to the question. So once we talk on the scientific procedure, to mention to outline 7 process of scientific procedure. The scientific procedure now, this is a way of studying things by testing facts systematically. So scientific method, this is a way of studying things by testing facts systematically. Tunakuwa tunasoma vitu na kuweza kutest uhalisia wa hivyo vitu systematically, meaning that this thing which we are studying, they needed to be scientifically proved. And that is one of the characteristics of the scientific experiment or any scientific knowledge. It must be subjected to approval so as to see its validity and the reliability. So it follows 7 steps which are identifying the problem. So these are the steps which you are needed to mention. So our answer this is just an answer. The identification of the problem, the first point. The second one is formulating of the hypothesis. The third one is experimentation. 4th one is observation. 5th one is recording of data. 6th one is data analysis and interpretation. And the 7th one in conclusion and reporting of the result. So these are the major scientific procedure. So this majibu yako ni haya, tu just wanted to mention like this in 7 points. Kwa wale ambao hajasoma, now let us be familiar with what is identification of the problem. Identification of the problem, you can, in order to identify the problem, meaning that you use your common sense. You have 5 major common sense. You have the eyes, you have the ear, you have the nose, you have the tongue, and the skin. So you use this sense organ to observe something. After observing something, now you ask the question. After asking the question, that is how you identify the problem. You observe, after observing, you ask the question. So identifying the problem, this is where a scientist make a puzzling observation. Observation can be made in the environment or from work of other scientists. So this observation, either you can use your common sense to collect the data from environment, or you can use the literature reviews from scientists basing on their findings to identify what is missing, what is the gap in that particular finding they made. So now you can come up with a problem and come up with the ways which you can use to solve the determined problem, and that is known as the formulating of the hypothesis. This is the second step in scientific procedure, which involves the formulating hypothesis. We are going to talk on the formulating hypothesis now. Hypothesis, this is a suggestion of the answer to the question asked. It is an intelligence guess that tries to explain an observation. For example, earthworm prefers moist and well-shaded area. So hii hapa hivyo, baada ya kuidentify the problem, unakuja na possible explanation, guessing. Unasema kwamba ili tatizo ni kwa sababu ya kitu fulani, au ili tatizo linasababishwa na kitu fulani. So these are the possible explanation or possible answer for the identified or stated problem. And this hypothesis can be either positive or negative. Lakini mara nyingi katika scientific research tunatumiaga ile positive, ile which can be identified. Null meaning that research anadili na kile kitu ambacho kiji. So if you are dealing with something ukifahamu, what you are going to do now? So you are dealing with the positive hypothesis. Another method is experimentation. After having your possible answers, alternative or suggestion for the identified problem, now you are going to perform the experiment. This is a science. It involves the use of the scientific proof to prove your answer, whether the hypothesis was true or not. You are going to test why this something happening. Example, you identified that you come up with the finding that the malaria parasite tend to live in this shady, poorly habited area, like those with uncovered pillar tree, with this of long grasses. So now you are going to carry an experimentation. You are going to collect your data by looking, is there any fact on what has been stated in my hypothesis? So now the experiment, this is a scientific procedure carried out under controlled condition to determine whether the hypothesis is correct or not. So this is scientific procedure now are carried under provided condition. This are called as variable. So in an experiment, scientific uses variable to test the hypothesis, and variable can be either dependent or independent variable. This is one of the features or characteristics of a scientific research. So the variable are condition or factor that can be changed or be changed, the example, temperature, speed, and light. So these are the conditions which are performed or which are used by the scientists to carry out their experiment. And another method is observation and data recording. After performing an experiment, now you observe what is happening. After conducting an experiment, you see what is happening. Now you observe, and after observing, you record on the finding that you have get from the conducted experiment. So the observation, scientists observe what happened from the time of experiment is set up to time it ends. It is important to note all changes of both dependent and independent variable. While observing what happened during the experiment and recording, make sure that you record each and everything that's happening after the experiment. Don't omit anything. Also, the result of the experiments can be recorded in a tabular form or graphical. So the data representation, the collected, either you can use the table, the tabular form, or the graphical form to represent the collected data. After collecting data, now you are going to perform the analysis and interpretation. Here you are going to analyze to see to what extent the collected data reflect to your suggested hypothesis. So the data analysis and interpretation, at the end of the experiment, the scientist analyzes the observation and the data recorded. Also, trend of pattern of data can be considered during the process. So this is data analysis. Tuna analyze kuangalia katika percentage gani kuna ukweli, percentage gani kuna uongo, au kama kuna kitu ambacho ni tofauti au imedeviate sana na hypothesis, na unafanya interpretation. Kwa nini hicho kitu, kwa nini hiyo majibu yamekuwa namna hiyo? The last one ni conclusion na reporting of information. This is the last process katika scientific procedure, scientific research. Baada ya kufanya interpretation, na unatoa conclusion depending on what you have conducted in your field of work, in the field. So unaandika conclusion kwamba hiki kitu fulani kimesababishwa na hiki na pia unatoa recommendation on what should be done. Na baada ya hapo unatumia njia mbalimbali kuweza kudisseminate your finding, that is reporting information. Either you can use the libraries, audio visual, video, ukatumia hata taarifa ya habari kwa ajili ya kuweza kutoa taarifa depending on what have been collected. Lakini sio kama tu unatoa reporting, must be first kwanza mpaka findings zako zikubalike na committee, the research committee wakubaliana nazo na kupatia go ahead kama unaweza kupublish your research work. So that is all about the process or procedure of the scientific work. And the question Roman B, you are asked to state why the handlines is useful in biology. The why handlines is useful in biology. This topic has been derived from that topic of the biological laboratory in that part of the biological apparatus. The handlines, this is one of the biological apparatus which is used by the scientists in performing a variety of biological experiments. So the handlines are useful in biological for different purposes, like magnification. Magnification, this is a process of magnifying this small specimen which cannot be seen by the naked eye, so that can be seen clearly. Kwa katika kukuza ndio magnification, vile specimen, kwa mfano kama ni mmea, kama ni tissue, kama ni any part of the animal ambao it cannot be seen by the naked eyes, kwa mfano hata protozoa cannot be seen by this naked eyes, tunatumia the microscope. Na vile vile inatumika sana katika field work, kwa sababu inatumika katika field work. Tumeangalia katika scientific procedure, kwa mfano unapoenda perform biological experiment lazima uwe na handless. Kwa mfano kuna baadhi ya structure ni portable kuweza kubeba, sio kama microscope. Unaweza kutembea na microscope unasafiri nayo. Unaweza kutumia handlens kuweka even you katika dissecting kit yako, ukaibeba handlens kwa ajili ya kuangalia observation, either katika kuangalia hata the wing venation of the insect or the bird. Ni vile vitu ambavyo uwezo kaviona clearly by the use of your naked eyes. Na vile vile tumeona katika identification of the characteristics. Kwa mfano kama fur texture, kuangalia texture ya fur, the fur, kwa mfano kuangalia unyoya, kuangalia ni smooth au ni maybe unaweza kutumia your skin as a katika observation, lakini si provide the correct answer, so that you can use this handlens katika kuweza kufanya identification. Vile vile kaangalia wind venation, the serration of the plant for those ambazo huwezi kuziona clearly. So this is why these handlens tend to be used in the biology. So just wanted to state the function of the handlens. And another question is question number 6 for the year 2017. And the question states that explain the following terms. Roman A, parasites. Roman B, host. And the last one is community. This is question 6 for Roman A. And the Roman B stated that give 2 examples for each of the following organism: endoparasite and ectoparasite. My lovely Form 2 students, these 2 questions has been derived from our lovely topic of the balanced of nature. Is where this question has been derived. It is a Form 2 topic. So for further information, also subscribe our media of Educare and search the topic of the balance of nature Form 2. You can get all the full packages pertaining this particular topic. Don't ignore, because you are going to get a lot of message. Also, is one of the very important topic. It tend to come from every Necta question. You must get the question from the balance of nature. So Now let us go to answer the question by defining the parasite, host, and the community. By starting with the Roman one, you are needed to state the definition of the parasite. Once talk on the parasite, these are the organisms that tend to live on other organisms. While they live, they tend to harm that particular organism. And the parasite, this organism, they tend to benefit while at the same time they tend to harm the host organism or the organism they live on. And that organism is known as the host. So the parasite now is an organism that live in or on other organism known as the host, and they depend on their host for survival. And the parasite can harm the host organism to varying degree, ranging from mild inconvenience to severe illness or even death. Example, Plasmodium protozoa ambao anapelekea hata kifo kwa binadamu akisababisha due to the excessive malaria, ambapo anaenda kuinvade the red blood cell pamoja na liver cell, ameweza kapelekea even the death to the person if not treated early. Vile vile another one is the host. Host ni yule organism ambaye anakuwa anahaba au ambaye anakuwa anambeba yule parasite. Example, human being is a host. So the host is this organism that provide a habitat and the resources for another organism known as the parasite. And host can be an organism supporting the growth and survival of another organism. Kwa hiyo hapa ndio host. Alafu the last one ni community. The community inasema this to the sum of the total of population of different organisms living in a specific habitat. So this is the meaning of the community. The question number 6, Roman B, need you to give the 2 examples of each of the following organism. The first one is ectoparasites. Ectoparasites, these are the organisms that tend to live on outside or attach on the outside of the body part of an organism or of the host cell. Example, kuna bedbug, kuna flares, kuna tick pamoja na these all organisms kazi yao, these are the blood suckers. They tend to suck the blood from the host cell. Example, animals, human beings, as well as the human being. Kuna human beings pamoja na other animals. Na kingine ni ectoparasite. Example of the ectoparasite, this one is ectoparasite. Second one here hapa ni endoparasite. This is endoparasite. And the endoparasite, these are the organisms, those are the parasites that tend to found within they tend to inhabit inside the host cell. Example, kuna hookworm, tumeangalia katika kingdom, mtasoma mtakapofika katika level ya form 4 katika kingdom animalia, katika topic yenu ya classification form 4 nimefundisha educare. Once unapofika level ya form 4, you are mostly welcome to subscribe and to get the full packages on the topic of the classification of living things for form 4. Kwa endoparasite example, kuna hookworm which infect the intestine of mammals including the human and leads to anemia and body weakness. Another endoparasite ni tapeworm pamoja na Plasmodium. Ambao tapeworm, this is example the flatworm ambaye vile vile anatend to inhabit katika intestine of the human being. Vile vile kuna Plasmodium, example Plasmodium malariae ambaye anakuwa inhabit katika female Anopheles mosquito ambayo ndio vector wa this Plasmodium. So when the infected female Anopheles mosquito anapokuwa ana take a blood bite to the person, it tend to inject this Plasmodium species. So akisha inject this Plasmodium, they tend to inhabit because these are the parasites and they tend to found within the body. They tend to inhabit into the body of the human being, where they tend to inhabit into the liver cell as well as the red blood cell. Once they reach into the red blood cell, they call the fracturing or the bursting of the red blood cell, ambayo inapelekea excessive bleeding leading to the anemia, inaweza kapelekea mtu akafariki. The last question is question number 7 for the year 2017, and the question stated that state the meaning of the term tissue as used in biology. Roman B, arrange the following terms in a sequential order from simple to complexity of cell differentiation. Umepewa tissue, organism, organ, cell, and system. Umepewa tissue, organism, organ, cell, and the system. Ya mwisho, outline 2 differences between plants and the animal cell. So this topic in general imetoka katika the Roman 1, imetoka katika ile topic yetu ya kwanza ya introduction to biology, and Roman B and Roman C imetoka katika topic yetu ya cell structure and organization. Both available at Educare Media. Just subscribe so to widen up your understanding. Without wasting our time, tukianza na Roman A, which need you to give the definition of the tissue. The tissue, this has been formed by the combination of the cell, and the cell, this is the basic unit of the life. So the tissue, this refers to a group or collection of similar cells that work together to perform a specific function. Example of those tissue, kuna epithelial tissue, kuna connective tissue, kuna muscle tissue pamoja na nervous tissue. The second one, umeweza kupewa this number of these cell structures, umeambiwa tissue, organism, organs, cell pamoja na system. Umeambiwa kwamba uweze kuziarrange kuanzia kuanzia katika simple to complexity. Tukiziarrange, anaanza ni cell anatengeneza tissue, combination of tissue inatengeneza organ, combination ya organ inatengeneza system, na combination ya organ system inatengeneza a whole organism inatengeneza population, ecosystem, biosphere. So lakini kulingana tulizopewa tunakuwa tunapanga like this. Inapatikana katika topic ya cell structure and organization. Just subscribe for further details. Nakuweza kujua what is cell, what is tissue, what is organ, what is system pamoja na organism. The last one, unaambiwa to outline the difference between the plant and animal cell. Plant and animal cell wanatofauti gani? The first one is plant cell have cell wall but animal cell have no cell wall. Also, the plant cell have chloroplast for photosynthesis, lakini animal cells is heterotroph, they do not have the chloroplast, therefore they do depend on other organisms for survival. Also, the plant cell have regular shape, lakini animal cell are irregular shape. Just one needed to give only 2 difference, so just make sure that you stick to the direction. Give 2 points, make sure that you give only 2 points. Up to now, my lovely Form 2 students, that's mark the end of an interesting and enjoyable session. Stay tuned at Educare Media for further updates on further learning topics here at Educare Media. Have a nice moment. Thank you for your attention.